And just briefly, didn't mean to cut off others. I mean, if there were any other things to bring forward before we get started. Nope, if not, well then, you know, in EBA, we're mostly devoted to student research and progress, but at the beginning of the year, at least once in the second half of the year, we like to development sorts of seminars. And so this is sort of a hybrid, you know, skills and career development seminar on reproducibility. And you know, Alden really is a wizard at this. I know that ever since your student days, you were way above the curve on this. And so we're very, very grateful to you for this.
Trying.
Yeah, but you know, trying and largely succeeding.
So as you'll see, I'm gonna show some slides from my dissertation organization when I was a student, and I'll let you guys judge how things were. I am gonna, you know, we're gonna talk about reproducibility. Feel free to break in and ask questions. If I get to the end of these slides without having to skip over other things, I will consider it almost a failure. So please interrupt with questions and stuff. So let's talk broadly about when we conduct a study, whether that study is an original research using data, or whether it's a systematic review, or maybe you do some sort of policy thing that I don't understand. Broadly speaking, this is what I would call the ideal way that we conduct research. We start with a study source. We get a data set that's either emailed to us, or we download it, or somebody transmits it to us in some cool way, shape, or form. You read your data into your statistical program of choice, R, Stata, whatever. You pre-process the data, you rename it, you generate your variables, reshape the data as necessary. You reshape it for your particular analysis, right? You select your records for analysis. More often than not, we're getting data from like a cohort study or a national study where we're not going to use everybody in that study, or maybe there's missing data on an outcome or something that we want to exclude these people. So this is its own concerted step here, determining who's in your study. The next step is your exploratory data analysis. Is probably one of the most important things you do. You make your table 1, do histograms, describe your data, understand why does 99% of my sample have a value of -9? Like, is that a problem? Should I be worried about this? Why does everybody in the sample have both the 10 item and the 30 item version of the Boston naming test? That can't happen. What's wrong with the data? I might have to go, well, regardless, you do your exploratory data analysis and you find all these fun and exciting surprises. I do a lot of modeling with M+ software, but basically the next step is write your model, execute your model, and get the output, and use that output to write your paper. You submit the paper for review, you deal with late reviews, and you publish in a peer-reviewed journal. For those of you who have written a paper, do I have it down? Does this describe things fairly well? I'm getting smiles and nods.
The only thing I would throw in there is that for statistical papers, you know, there might be a slightly different set of things, say from 5 to 6.4 if they're simulation based or whatever, but you know, that would be the only reason.
You develop your simulations and then you evaluate diagnostics of them.
Yes.
Okay, yeah.
I'll also put an arrow back to 5 from 6.4. There's always a circle, like
Yeah.
several rounds of analysis.
So you're anticipating what I'm gonna say next. This is the ideal way to conduct research. This is what I would say is the reality of peer-reviewed research. You read your study data into your program, you get your data, you get your source data, you read your data into your statistical program, and then you pre-process. And as you pre-process, maybe you realize that you got this data from an Excel file that was created in Italy, and Italian versions of Excel have this hidden character that make 90% of your data missing. And you didn't realize it until your pre-process, actually, in my case, I didn't realize it until I was doing my exploratory data analysis. Weird things can happen, especially with Excel, even CSV files. So understand that you're going to be going in a lot of loops. You're going to be doing, especially by the time you get to exploratory data analysis, who's unmuting? You're going to be running around a whole lot trying to figure out what's going on here. Eventually, you're gonna do your modeling. There is a circle in here for troubleshooting. And I can imagine, especially with simulations, you've got to troubleshoot in lots of different ways. Did I set up my parameters in a believable way? Do I have to do sensitivity? What's the difference between a sensitivity and this analysis and the secondary analysis? Eventually, you do get model results. We all get results from our models, and we make our results tables, and we write our paper, and we submit it for review, but it really just takes a lot of time. And so you might want to think about getting a hobby while you're waiting. Eventually, our papers do get published, but it takes a lot of time. And especially between the time that you submit a paper for review and the time that you deal with comments from your esteemed peer reviewers, who you may never know, you may have moved on to other projects and other things, and you may have forgotten all of these nitty-gritty details that you did up in the pre-processing and how that affected your modeling. So there's a lot of things in every study to keep in mind when we do it. And as you go forward in research, we accumulate more studies, we learn more things. So I had the fortune of writing an exploratory data analysis of a graduate student that Liz Stewart suggested that I wrote this paper in between the time when I submitted my final thesis for Karen to review and when I had my final defense. And this is one of the figures from that paper. I highly recommend the paper. It's funny.
It's in Chance, right?
Yeah, Chance magazine. Yeah, somebody published these things and they got peer reviewed. But anyway, this is inches. I went back to 5th grade. That's when I started managing my own papers, and I just measured the amount of stuff I had by year. Everything was organized by year and by subject, and until 9th grade, there's not much going on because, you know, drawing pictures of things. But starting in high school, you see this exponential curve. In college, you see this exponential jump. The more amount of time that you spend in a place, the more stuff you're accumulating, the more you're doing, the more you're producing. Graduate school, we see a dip. That's only because this is the late 2006 to 2010 timeframe when we're really starting to be more conscientious of the environment. For those of you who don't know, when you take Biostat 620s, you get like, I don't know, a website or a USB drive with all of the lectures in PDF form. It used to be that they gave you binders. Each term, if you were to print these out, you would get binders this thick of all the lecture notes.
We got the binders, they were printed out for us. I still have.
Yes, I still have mine too. It's underneath my desk. But yeah, that's why you see a decline. If you were to look at megabytes on a USB stick, which I don't have it, it's in my bag. You would see an increasing function for megabytes of file sizes, but that's fine. So how do you manage this fire hose of information and data? Well, you know, so there's a whole area of research around reproducible research, and I picked a paper that I think does a good enough job for the point of this presentation, but I want to acknowledge that there's a lot of people, like I'm thinking of Roger Peng, have devoted a large portion of their careers to reproducible research. There's a lot of papers out there. This is just one drop in the bucket. And I kind of like the 10 points here. So what does this guy recommend? So for every result in your paper, keep track of how it was produced. For the most part, most of your numbers and your regression coefficients, your descriptive things are going to be in tables, but there might be just one thing that doesn't need to be in a table, so you just put it in the text. Make sure there's a record of how it was produced, especially when after you get those peer reviews from somebody, and they'll ask, well, you reported you know, a sensitivity and specificity, but what about the area under the curve for this relationship or something? Okay, we can go do that ad hoc, but make sure you document it. Avoid manual data manipulation steps. We mistype things and make errors all the time. The title to this slide, I misspelled computational. So, I mean, I'm going to show some stuff near the end that shows even like reproducibility is something that I aspire to, but I make a lot of mistakes. We all do. Try to automate your work. I'm going to show you a type of programming in this talk where in an ideal world, you could open up one do file and click and run all the components of that do file, and that do file is going to call other things, and you'll end up with a nice PDF with all of your results and a nice proverbial bow, and it's all automated. So figure out, and you can automate your work, and by that I mean don't hard code variable names, don't hard code, avoid hard coding as much as you can.
Define hard code.
So I might use a variable U101. Or I may hard code, do a certain thing with a value with observations for people age 73 years of age. Try to avoid that if you can. If there's something you wanted to do about age 73, instead of referring to age 73, maybe you're picking on age 73 because it's your minimum age in your data set or your maximum, whatever, or it's, so just refer to the minimum age in your data set, not to age 73. Or don't refer to U101, refer to the first variable in this particular data set that begins with the letter U or something like that. Try to avoid hard coding and automate stuff as much as you can. Archive the exact versions of all external programs used. Number 3, this is kind of a dig at R software. So when I was in the 750s taking Epi 750s taking epidemiology, I was in a lab group, and we were in my lab table was our first term. So we were doing something with regression. We were looking at a regression output. And another lab table had a slightly different result in a way that didn't matter, but it was the numbers, the regression beta coefficient was different by like in the 4th decimal place. The reason for that was a versioning difference in R. And yeah, so something that's different in the 4th decimal place probably doesn't make a difference, but sometimes if you're talking about that difference between p equals 0.05 and 0.049, it can suddenly make a big difference what version of a program you were using. And as it turns out, we're dealing with what could be a high profile paper right now in the active study. I'm asking a question about cognitive training and dementia, where table 3, the confidence interval does not include 1 for an odds ratio, but table like 15 in the supplemental materials, which has interactions, but in theory, it should be the same table. It just barely includes 1. It's like 0.99. So we have to go back and figure out what version of programs was the analyst using for when we were running these things. Version control your custom scripts. So you might run something, you might do your exploratory data analysis before you run your models. You might redo it after. You might do a new set of do a new set of pre-processing after you get reviewer responses in step 8. So just keep in mind, just version control all of your scripts when you can. Record all intermediate results when possible in standardized formats. For example, you might have the raw source data set, and you might have a final data set that you use to run commands and estimate your model output for your final paper. But before that, you selected cases for analysis. Somebody might have a question about what was the raw data set, what was the original data set, and what happened after you made this exclusion. Or something like that. So record all of your intermediate results. For analyses that include randomness, note what your underlying random seed was. And this is something that I'm beginning to struggle with. Apparently, different programs can have multiple underlying seeds. So I know how to hard code a seed. I always use the seed 8675309. And it's really heartening when I give somebody a program and years later, I'll get something back from one of their trainees or from them themselves, and they haven't changed my seed. So that's really cool. I'm excited. But note your underlying random seed and understand how randomness is happening. Got my number. Yes, I do.
I wonder if most of the people in this room are too young.
They're probably, yeah.
86750. There was a song.
So.
You guys have Tommy Two Tones? Yeah, God knows Tommy Two Tones.
Okay.
Always store your raw data behind the plots. So when you make a histogram or when you make, say, a line plot showing point estimates with standard errors, whatever you've made, there's some raw data underlying those plots. Even for something that you think is as simple as a histogram, it matters how the rendering of that histogram, it matters how wide those bins are. You can make a normal distribution look uniform. You can make bimodal distributions look normally distributed by messing with bin size. So make sure you store the raw data behind the plots, which is kind of goes without saying, as long as you've recorded the exact command that you used to estimate a thing. Generate hierarchical analysis output, allowing layers of increasing detail to be expected. So at one end of the spectrum, you've got a raw data set. At the other end of the spectrum, you've got a published paper. Let's say you published your paper, but maybe there's supplemental materials for this paper. But even beyond supplemental materials, even the most picky journal won't want necessarily your line-by-line analysis output, but not everybody wants that. So think about how you would generate hierarchical outputs of analysis for people to ask increasingly, to increasingly scrutinize your results. Connect textual statements to underlying results. This is kind of like step number 1 here, I think. But any time you say something in the text of your paper, or if you include something in a report, make sure you're clear and that you comment your code. Make sure you're clear how you got there. And provide public access to scripts, runs, and results. I think this is increasingly, fortunately, becoming a standard, especially in gerontology and within a lot of fields that we work in. GitHub is really common, and you can also, if you want to, you can publish model syntax and supplemental materials of your papers. That's really cool. It's still a little difficult to record or to publicly share data oftentimes, because many times as the person writing the paper, we don't have the say to say, I'm happy to share my data. Maybe I'm dealing with geographically privileged data or Medicare information or stuff that is behind some sort of a wall that requires extensive DUAs. So data sets are hard, although sometimes it's possible, but your syntax should be readily shareable, I would think. So consider providing public access. And it's been shown that people who are more open, who share their syntax and this interim stuff, tend to be more widely cited because junior people and grad students are actually have their material and they're replicating them. And so, oh, I'm going to cite this person because they gave me their syntax. That's really cool. So it's better for your career as well. Pablo?
Yeah, I haven't shared my code yet in a paper that I published, but I've been in a couple of labs where they ask that you share the code with one of your co-authors, and they're going to make, like, to gain their co-authorship, one of the things they do is reproduce everything. So
Yeah,
and that was very humbling to me. Like, you have to work much harder when you do that.
so code review is a whole other topic. Like I said, reproducible research is a big field.
Yeah.
So there's a lot of people I'm sure have opinions on how you do code review. I was just talking last week with somebody who wanted to describe in words what his syntax was doing, and then have another analyst on the team implement the code as described in these words. I think that's measuring two things. That's measuring not just coding, that's measuring one's ability to communicate. But when you run code, do you just simply run somebody's analytic file, or what do you do?
In that case, it was, yeah, basically just rerun it, but making sure that the line, like, everything makes sense and is what you have in the tables and all that.
There's a continuum
Right.
of things, and I don't think I have a good answer. So where do these norms for reproducible research come from? I learned over the summer from Ted Miguel at Berkeley. His face is a little fuzzy. That's okay. He's not fuzzy in real life. But a lot of our norms come from Thomas Merton. So Thomas Merton wrote this book in 1942 that you can buy on Amazon for like $45. The guy died in 1965. I'm not sure who you're paying $45 to, but I digress. It's an influential articulation of the norms and ethos for scientific research. These Mertonian norms, so to speak, are grounded in an understanding of scientific research as embedded in social structures and institutions. So it depends a little on your philosophy, but from a social constructivist perspective, there's an argument to be made that everything we do is driven by social needs and the needs of society, and there's sort of a handshake between society and government funders and researchers, us, as to what's important, what's relevant to research. Alzheimer's is important because Ronald Reagan had it, and my grandparents are affected by dementia. So let's spend a lot of resources on dementia. People have problems with walking or with frailty. So this is something that I want to study. And oh, look, I just got a grant from the NIH to research this or that. So okay, somebody else, society agrees that we should study this. A set of norms are binding not only because they're procedurally efficient, but because they're believed to be right and good. They're moral as well as technical prescriptions. And so Mertonian norms are also where we get the notion that you should consent people and all this other stuff. But I'm really gonna focus on the second of these four core values. One is universalism, and then communism or communalism, which is the communal sharing of ideas and findings. We believe that that's important, especially in our research as scientists. We want to share what we learn with the world so that we can advance science advance the world's understanding in an incremental fashion. Disinterestedness, which is really interesting for another day, and organized skepticism. Yeah, so I thought that was interesting. I haven't read this book, but I want to someday. Maybe somebody can tell me how it cost $45. But anyway, I've got some quotes here. The substantive findings of science are a product of social collaboration, especially for us who are NIH funded, because public dollars funded this research. And the findings are assigned to the community. There's an emphasis on open sharing of scientific knowledge. Secrecy is the antithesis of this norm. Full and open communication is its enactment. And the community of the scientific ethos is incompatible with the definition of technology as private property in a capitalistic economy. That's why this one's called communism. There are different norms for if you're in an academic versus a corporate or private setting, although the distinction over time has been eroding. You know, a lot of pharmaceutical and medical device companies, and also I'm thinking about genetics companies, they still keep a lot of their findings and methods under wraps, but increasingly, you know, if you're a biostatistician at 23andMe, if you get proper approvals, you can publish a paper if you get proper approvals. So it's possible. So some needs for reproducibility. This comes from that Solvay paper, the 10 recommendations for reproducible research. We need a file management system, and we need to version control our analytic scripts and our other documentation. We need a file and variable naming convention. We need logging and documentation, and we need data dictionary functions. These are super important. And you want to make sure that you archive your projects. I think in my brief career, I'm on my 4th or 5th laptop. And if I were living on hard drives, I would be having a real problem here. But fortunately, I don't. I'm gonna cover at least 4 out of these 5 things. Reproducible research, at the end of the day, it makes life better for you because it makes your life easier. It makes life better for your colleagues who you're going to give some of this stuff to, and it's ultimately better for science. I remember once when I asked one of my doctoral students if she could send me her code that she used to do models for her dissertation. This was early on, and I thought, oh, I'll do a code review. I'll do a buddy check. She sent me a 5,000 line do file. I noped right out of that. That was going to be hard. The problem with reproducible research is it's hard to be reproducible. You know, so I do a lot of original analyses where we have an exposure and an outcome, or maybe we have a measure that we want to develop and we want other people to use this measure, but we have to establish its validity and reliability and stuff like that. But somebody else may be doing a systematic review, and somebody else may be doing a simulation. So how do you distill original research projects into distinct common elements? It can seem arbitrary at times. There's a spectrum from liberal rules to strict rules. Liberal rules are what I would describe are easy to set up. Let's just make this folder for, you know, October 9th, 2023. I think it's the 9th. And this is what I was doing on this day in this project. And then you can have stricter rules, which are harder to set up, but they're easier to remember in the long run. So in 2030, when I go back, I don't have to magically remember what I was doing on October 9th, 2023. I can go into this posted analysis project folder, and I can pick up from where I left off. There does exist a generalizable workflow approach that follows strict rules, but it's fairly easy to set up. So you get the best of both worlds. It's great. And I'm gonna talk through that today. And fortunately, it's something that I didn't make. But first of all, let's talk about something that was set up with liberal rules. My dissertation folder, Karen. This is the parent folder over on the left here. And, you know, administrative, this makes sense. This is where I kept minutes from meetings with my advisor. Analysis results. Okay, which ones? There were like 6 chapters of the thing. Archive, well, I don't know what archive is. Graphs. Okay, well, graphs are over here. There are graphs from November 9th, 2009, January 20th, 2010, and so on and so forth. And then there's graphs named 1, 2, 3, 4, 5. AIC, I think that AIC combined, I don't know what these are anymore. I think I know what manuscripting was. That's when you're pulling apart your dissertation and you're writing papers for it. M+, well, that's a software, but what was I doing in M+, and how is that different from analysis results? NIA data sets is one type of data set, but you'll notice in the raw folder, I've got all these other data sets. Sometimes it's called November 16, 2009 strat with an N of 1401. How does that differ from the N of 8406? Why am I saving a data set that's called imperfect? I don't know. Why is there a folder called fun? What exactly were we thinking when we made a subfolder called fun? As it turns out, fun is where the exploratory data analysis of a grad student came from. But we can go on, and it's just a mess in here. And then we have strict rules that I'm going to talk to you guys about today. This is a textbook published by J. Scott Long called The Workflow of Data Analysis Using Stata. J. Scott Long is a professor at Indiana State University. I have no idea what research he does, but this is a really cool textbook. And I'm pretty sure it was at one point, and I'm pretty sure it's still a recommended textbook if you guys take the Stata programming class. So it's a good book. He goes over elements of common workflow organization, which includes basically a folder structure, or a structure for folders for your particular project, as well as analysis files. How you divvy up your analysis into distinct files. Long's advice is actually to never have a do file or R script file that's more than like 10 or 20 lines. Make it small and incremental. I don't do that. But I at least try to differentiate between calling a data set and pre-processing your data set and running your models and stuff like that.
I mean, so all for way back in the day, that would have been the subroutine driven way of programming, right? To drive things up into, you know, sort of discrete elements and then pull those elements together
Yes.
in a master file.
Yeah, exactly. And I'll show some examples of that. This is the common folder structure. So I have on my computer a folder for C work, and C work has dozens and dozens of subfolders. One of them is Repro Research, and Repro Research has a set of folders that are constant across everything that's in C work. So we have an admin folder. In the admin folder, you can store information about the budget stuff about your project, correspondence, IRB approvals, proposals, if this is derived from a grant that you wrote. If you didn't write a grant, and if you don't have anything to do with the budget for a project, if you only received a data set, and as a student, you're told to analyze this data set, that's cool. You don't have to do anything in admin. But this is like an oversized shell for this folder structure. It's like an oversized shell for a turtle. Not all projects need to grow into every folder and subfolder, but this can grow to encompass large R01 type projects. For something like the ECA, Adam, I would recommend multiple maybe analysis or project specific folders. So just to give you a frame of reference. There's a documentation folder at the same level of admin where you store things like your code book or Q by Qs, the actual questionnaires that this study administered that you're using the data for. The posted folder, as an analyst, this is where I spend most of my time. And there are several important subfolders in here. There's posted analysis. In the analysis subfolder is where you store all your analytic files. These can be Stata or R or SAS or what have you. Then you have your data, and your data always comes in two types. There's either your source data or your derived data. Your source data is some sort of data set that you got from a source. Either somebody emailed it to you or somebody has or you downloaded it from a website, or you got it from something official. Now, in that source data set, the only thing you ever do is open the file. You don't change a label, you don't resort, you don't change a variable from string to numeric. Don't do anything. Leave it raw. Even if it's an Excel file or something. One of the most annoying things is when a non-data person gives you an Excel file and they put descriptions of what to do with it at the top, so that the actual data only starts at row like 11. Well, don't change it. Because guess what? This person might update it. Just plop it into source and write code in your analytic file in the call source do file, write code that only starts reading after line 11 of this particular sheet. That's fine, that's doable. Anything that you do edit, any changes you do make to a data set, resave it in the derived folder. And you can have subfolders within derived as needed. But there's a lot, there can be a lot of things going on in the derived folder. There's a subfolder for descriptive statistics and figures. There's a text folder where you can store those interim reports to allow hierarchical levels of analysis output that we recommend in reproducible research. And then you've got a private folder. So for any given project, there might be a particular do file that you received from somebody, or a paper draft or an abstract draft that you're working on, but maybe you might be sharing this folder. It might be in a shared location, like in a OneDrive or a Box folder, where you can be sharing this folder simultaneously with somebody else. So in my imaginary, well, not imaginary, this does exist. In my reproducible research folder, I have subfolders for AS, JAD, JAS, KBR, and QLX, and all my friends, and they're welcome to add whatever they want to this private folder as it relates to the project. You should not store your taxes in your private folder because it's not really private, but it's your own play area. The cool thing is in Stata, there's the thing that makes this hard and strict is you have to set up these folders, and so you have to right click, new folder, name it this, right click, new folder, name it this. There's a Stata ADO that basically creates a batch file that creates this folder structure for you, and that's WF tree for workflow tree. And so this is example syntax that would make that for you. That's fun and easy. So that's a folder structure. This is the common folder structure.
And Alden, before you go on, let me just briefly ask the people, who use R frequently, and who would have been taking the data science courses and that sort of thing. Does this resonate? And is there a similar, R structure that you learned, or I see one, not as much, yeah.
I'll just make my own folder if I work with R. I mean, even with Stata, I just make my own folder of the structure.
So what if you're working with other people and you want this person to work on massaging the data while you work on the modeling.
It hasn't become a problem for me. I'm at the bottom of food chain.
Yeah. As you get up in the food chain, you're gonna be working on these collaborative projects where where different people might be in different parts of the program at the same time. Like I'm working on a project right now where we're doing cross-national comparisons of cognitive functioning, and somebody has to fix the education do file for calculating education in the ELSA study in England. Somebody else has to fix the cognitive variable processing in the Indian sample. And people can be opening these different analysis files at the same time. So we're on a shared platform of some sort. In R, there is a way to refer to the working directory and then one level up from the working directory, and then you daisy chain it, move one over to the next subfolder down in the working directory. So in that case, you wouldn't need to refer to a derived folder or a source data folder. So yeah, there are ways around this. This is just the approach that I use. So that's a common folder structure. This is a common set of analysis files. And like a large think about a shell of a turtle. There are 14 common files, but you'll notice that by the numbering system here, these are ordinal numbers. We can add more do files as needed to expand as needed. So first of all, there's an ADO that writes all of these do files for you called AutoMaster2, and here's your syntax. And the cool thing about AutoMaster2 is not only does it create these do files, there's certain common text within the do files that it also will write. So it's automatically going to save at the end of your call source. It saves a data set called interim 105 call source, and it saves that for you in your derived folder. At the end of 120, select your cases for analysis, it saves that data set in your derived folder. But broadly speaking, there's a master do file. The only contents of master do file are the names of all these other do files, and you can use the include or do function to run all of these other do files from the master. There's a preambling statement where you set your seed of 8675309, and maybe if there are local programs that you put together that are specific to this project, you can put in those little macros yourself. I use LaTeX. LaTeX is similar to R Markdown. It's something where you can weave in results and analysis file and analysis things and remarks about the data as you're coding and as you're writing your code. Call source. I don't care how simple it is. It could be one line, but put it in its own do file. It could be one line or it could be 100 lines in the case of my Italian Excel file. Create variables. This is where you create and pre-process variables, you rename variables, you label the values or label what the variables are in some way. Anytime you're massaging variables. And then the next step is select your cases for analysis. As we talked about before, you usually want to select a subset of your cases. There is some give and take I found between what I'll call 110 and 120, because let's say that part of creating a variable is standardizing it to have a mean of 0 and a variance of 1 in your sample. Well, it's okay, so it's got a mean of, this variable has a mean 0 and variance of 1 in the sample of 3,491, but then I dropped 491 people. So the mean and variance are no longer 0, 1 in the sample of 3,000 people. So you might have to redo a few things here and there, but just comment things and make it clear what you're doing. After you select your cases for analysis, you're ready to do exploratory things. So make your table 1. your table one across most projects is a descriptive table. It may not be true of every project, but even for a systematic review, you got to describe the characteristics of these studies that you're using. So make your table one, and maybe it's got one column of statistics. Maybe you've got your exposed and unexposed columns for those. That's fine. Figure one is often some sort of a distribution or a description of the main outcome before it's adjusted. You don't have to. Oftentimes this doesn't end up in the paper, but it's nice to put it there. And let's have its own do file for it. You'll notice that the number here is 210, and then we jump up to 305 because 305 is the model statement. And as epidemiologists and as biostatisticians, this is where we shine, right? We love to run our models. So of course, you know, as Long would say, do one model per program, or you can, I find you can, if you're running like 30 models in a loop, you can have that one loop in one program. That you don't have to be that crazy. But you can have multiple analysis files, multiple do files for different models. Just be clear about what models do what's doing what. Ultimately, we have a table 2. These are results from your models. Maybe you've got a table 3 and 4 and 5. That's fine too. So, you know, just we go from 310 to 320. You can add those 2 files. Maybe you have a figure 2, which is model implied results. You're going to definitely have a lot of sensitivity analyses, both before and after you get to reviewers. So, you know, this is one sensitivity analysis do file, but you can always make more. At the end of your analytic run, you might want to write out some conclusions or next steps. So stuff in text. This is stuff you might want to write in text. All throughout all of these do files, you're at least for me, I'm weaving things into a LaTeX document that I'm then going to make into a pretty PDF and save into the posted text folder with a date stamp. And then we close LaTeX and run it. There's an ADO that writes these do files for you in Stata called automaster.do. I think I already said that. Okay. You can initiate your workflow on the fly. There's an ADO for that too. So the key to this, to my flavor of workflow, is all of my folders are stored in C work. And so I just have to go to my C drive and then to work, and then I can select what project I'm working on at the time. But as long as I'm using a common folder structure here, the only thing that changes is the name of the project, C Repro Research. I could go into my ERIC folder or my BioCard folder or my HCAP folder, and all of them will have a posted analysis folder. All of them will have a posted data derived and posted data source folder. So there's an ADO to initiate my workflow for me. I'm a Windows user, and I recognize that things are different for Mac users. But you know, there are at least folders on it. I just don't know how to use them. So I do recommend weaving in a document writer for reporting, either LaTeX or RScript or whatever the next popular thing that's going to come out in 5 years. Here's some examples of some workflows. So like I said, this at the left-hand side here is my work folder where I have folders mostly named after data sets, although I'm starting to evolve beyond that because you'll do multiple studies within a data set. And you don't want to necessarily have multiple papers or projects going on within one folder. You'll confuse yourself, as I have. There's a subfolder for the MoCA. I was doing a MoCA analysis once. There's a subfolder for BioCard, a subfolder for ERIC, which I've been getting rid of because ERIC is moving to a shared design.
Dagobah is an intriguing folder.
Dagobah is kind of a swamp of do files
Okay.
and analysis files that I share with my students, like if I want them to learn about the scatter I command or PC spike in Stata, I'll throw in some example syntax there and they can play with that. They can do whatever they want.
So question,
yeah.
so you have different iterations, like you run things over and over again. So each time you'll create a subfolder with a date that follows the file structure, or you just like overwrite?
So so if I do an analysis, actually, I'm on the internet, yeah. Let me show you a thing that I was working on. So I was trying to make a graphic yesterday. And there were a lot of fits and starts with it. And it started out looking like this. You don't need to store this as your folder, as your you don't need to store the code that produced this. This is a triangle. It's a red triangle. This is looking a little better, okay, but it still doesn't have labels, still not clear what the heck it is. This is nice. It's got colors. You can see that there are different columns. It's starting to look like a map of associations, but the labels are all messed up. And then eventually, this is getting better. The labels are still the labels on the left are still messed up. There's a spacing issue. You don't have to store interim results for every single aborted figure or program you make, but anything that you share with somebody, anything that you're thinking about maybe publishing, you should store that. And if you make a slight tweak, like if somebody can ever explain to me how I can increase the spacing in Stata between these rows, I'm not going to make a separate do file for that, but I am going to make a note, like, you explained to me how to fix this silly thing on such and such a date. So I fixed that. So I toggled that.
And then Alden, as you're getting back, we're getting close to the end of time, and so whatever is the most important message or two to the.
Okay, so this is so I'll go forward. This is thank you for that. This is the Mocha workflow. So within C work Mocha posted analysis, there are a bunch of do files, and this I've grown a little bit. There are a number of sensitivities that we've done. This is what the master.do looks like, where we call other do files. This is an example of that hierarchical analysis input. I've got a PDF report dated September 3rd, 2015, and I've got other reports going back as far as 2013 that describes the background of the project as well, and then the analysis plan, and then the rest of the report shows bland Altman plots and regression outputs and all that fun stuff. And this paper was dead in the water in 2015. If anybody would ever like to pick it up, I will send you this report dated September 3rd, 2015, and ask you if you'd like to make this your master's project or something like that. And if you say yes, then I'll send you the rest of the stuff. But hey, you've got the results as they exist in 2015. This is my biocard folder. This is a more well lived in folder, almost irresponsibly so, because there have been multiple papers and multiple studies published in the biocard study. So there are subfolders for biomarkers, for CCFNQ, which was a project that I was confused about. There was a project that I was working on with Zheyu Wang in the biocard study. Each of these subfolders have their own workflow analysis folders. And here's another report. This was the role of inflammation in cognitive impairment that we wrote. This is a report in 2018. This 2018 paper became a 2018 report became a 2019 paper, which is totally cool. And as of late 2023, it turns out we found some interesting results. And so if it's interesting results, society says you should get more funding and assay more people and add more follow-up time and include more additional biomarkers, because this is a really groovy finding. And so we have to reproduce our results. So it's important that we kept our analysis files and our data as they existed back in 2018, because it's 2023 now and somebody cares. This is an example of an admin folder that's well lived in. So Frailty Cog was the folder structure for my K01. And so at that point in my career, I was helping people out with grants. And so I worked with, say, Peter Abadir on something. I worked with Michelle Carlson or with Mara McAdams, or with Karen, we wrote an NIA methods proposal. That instead of creating separate folders, this was in the admin folder of my K, because I thought these were K related. That's fine. It's the notion of it's important to maintain records. This is an email I received in 2017 asking me, I understand if people need to leave, asking me for data and results from 2011. So that project is still ongoing. So yeah, you want to make sure that you're maintaining your results in a way that you can remember. This is the paper from 2011 that she's referring to. So these are our 10 guidelines, and they're really important. We covered these aspects, and so some key take homes. Never ever change your raw data. Use an automated program that minimizes data entry error. Never ever change your raw data. Building redundancies and error checking into your analysis files, and never ever change your raw data. Most importantly, help each other, help your colleagues, ask for advice when you need to. Bonus material, this is what I'll end on. The importance of not copying and pasting. So I published a paper, it came out last Thursday in Lancet Healthy Aging on the harmonization of later life cognitive functioning across national context, results from the harmonized cognitive assessment protocols. It's funny that the English decided to misspell harmonization here, but not here. But who am I? This is fine. And so this was the folder structure. So with this paper went through 3 rounds of revisions between July 26th and August 17th of this year. And during those times, I was working on revisions on planes between Amsterdam, LA, and Lake Tahoe and home. So you'll see certain folders like last lap. That means I think this is the last time we had to do this. But like there's revisions 1, revisions 2, revisions 3, signatures for stuff, proofs, but I digress. This was table 1 after it went through co-author review and after it went through peer review, through 3 rounds of peer review. And if we can pause for a moment and look at this table, this has been reviewed and re-reviewed. Any issues you guys see? I'll wait for you to pick out just one.
The first column has percents for gender, and the rest of them don't.
Oh, that's a good point. Yeah, we wrote this down. I'll tell you something else that's concerning. This is male and female. Do these numbers add up to this number? No. What about these numbers and this number? What about these numbers and this number? Are you getting nervous? Go authors, you were supposed to read this thing, guys. Fortunately, if you guys have never published in a Lancet paper, they rewrite your paper and they check every single number and they recalculate your percentages for you. This is after it got through the editor. As it turns out, between the Charles HCAP and the Halsey HCAP, I transposed the columns for education. Between MEXCOG and La Ciudad, I transposed the columns for male and female. They want, it's a matter of policy that for any variable, they want an extra category for missingness. So they wanted me to complete a missing this row. Note there was no missing this row for education, but about 30 or 50 people are missing it in the ELSA HCAP sample. This is terrifying, isn't it? This was supposed to be re-reviewed, but somewhere on an airplane between last lap and matter for submission of revision 3, I gotta admit, I was no longer, subscribing to my rules of don't copy and paste and automate everything. I was just like, get this thing in. Like, they have rules about instead of percentage number in the text, they want to see number and percentage. And they're very particular about things. So yeah, imagine even if you aspired to be reproducible and to adequately code up your data, if this was the extent of a problem in table 1, what are some of the other problems that might be in papers from other journals too? Maybe, hopefully nobody's misspelling words in the title of your paper, but So in terms of dissemination, this flavor of Long's workflow is being used at lots of institutions and lots of groups. It's been adapted here at Hopkins by an adaptation of it by the ECHO Consortium. And Long's textbooks is optional reading for the State of Programming course, and it's available for free on the Russian websites if the Russian websites happen to still be around, but I didn't say that. Thank you.
Thank you. And so I do want to absolutely put out there, anyone who needs to go for overtime and, feel free, that you can go on. And what I would say is for others who can stay or want to stay, why don't we take, just a few minutes so that then we can get ready to go, to the happy hour. So we'll take, until say 4:45. So let's open it up for any questions.
Jamie Smith asks, how did you get a screenshot of my files?
Yeah.
Don't forget version final.
Oh, I see Lacey has raised her hand. Go ahead, Lacey.
Okay, Lacey, yeah.
Oh, I was wondering how you back up these file structures that you create.
So I have an external hard drive that at random occasions I'll back up. It's a good idea to get used to GitHub though, because with GitHub you can automate the uploading of files too.
But you couldn't, for example, upload like a lot of sort of the data and, the.
Not necessarily to GitHub, but if you've got a hard drive, if you could back up the hard drive, or if you've got you can use shared spaces. So Hopkins used to allow Dropbox, but now they don't allow Dropbox, but University of Michigan and other institutions allow Dropbox. Hopkins used to have JHBox, which they don't anymore, but everybody else does. Hopkins makes it really difficult to share with other people outside the university. Now we use this Microsoft product OneDrive, which I don't know how to make it work, but in theory, you could make OneDrive work.
Okay, thank you.
Yeah. Michelle asked, could you speak to the use of Eric now that Joe is moving to NYU? That's a really good question.
Meaning what, like how we're getting the data or?
The data will still be stored here. You're still going to be able to use it and using their remote enclave thing, I think. Jennifer Deal says, I recently found an error in a Lancet Public Health paper after it was published. Numbers in table 1 didn't add up.
Ooh, was it all this? I know, I have a theory about why they didn't change on your, will you show the title of the paper?
Oh,
Which slide is
yeah.
that? So I think because the harmonized versus harmonized is because harmonized cognitive assessment protocol is the title of the study.
Yes, it
So
is.
there harmonizes with an S, but because that's your title, that's why. That's my theory anyway.
Okay.
Yeah.
Maybe, yeah, I could see that.
Well, with that, why don't we call it? And, Alden, thank you so much once again. That was fun.
Thanks for listening.
All right, well, so hopefully see some of you down at, as many as possible in the Ministry of Brewing soon.